So before we get started, I just wanted to make a brief introduction to the BioFlux system for those that are not familiar with it. The BioFlux system enables high-content live cell assays in vitro. The system emulates physiological microenvironments by controlling factors such as fluidic shear stress, compound perfusion, temperature, and cellular invasion and migration conditions. The system is comprised of a microfluidic well plate device, a pneumatic control instrument, and a range of hardware options for running live cell microscopy experiments. The system is optimized for microscopy. It can be used with any existing inverted microscope or combined with a turnkey BioFlux 1000 system for advanced automation and convenience. So I'd like to now introduce Jo James from the University of Auckland to get started with her presentation. I'm just going to, let's see. Okay, Jo, are you with us?
Yes, I am. Hi.
Great, thanks. I'll let you get started.
Okay, thanks. Hi, everyone. So thank you very much for tuning in today. Where are my slides, Mike?
You can just advance one slide and yours will be
right,
next.
Okay. There we go. So thanks very much also to Fluxion for giving me the opportunity to talk to you about my work today. I've recently just moved back to New Zealand, to the lovely Auckland. But today I'm going to talk to you about some work I've been doing for the past few years over at St George's University in London. This has been looking at the effects of shear stress on uterine vasculature remodeling by trophoblasts, which are cells in the placenta. So feel free to jump in with questions at any time you like, and I will try to address some of them as we go along if I can. Okay, so for those of you who aren't too familiar with the placenta, this is the fetal organ. So it belongs to the baby, and it forms this interface here between the mother and the baby throughout pregnancy. So this is really important for pregnancy because all of the nutrients and gases that are exchanged between the mother and baby have to go across the placenta. Now, there's another, sorry about this. There we go. Okay, so during pregnancy, the placenta has a villous kind of tree-like structure. I'll just jump back to the previous slide, sorry. And so this is surrounded by trophoblasts, and these trophoblasts grow out of the placenta and invade into the mother's uterus. So this acts to physically connect the uterus to the placenta, but it also remodels the uterine vasculature. So it takes the tightly wound spiral arteries in the uterus and converts these into wide-bore conduits that are completely independent of maternal vasoconstriction. So this allows a constant and increased supply of blood to feed the placental surface and the baby as it grows. And we know this process is really important for pregnancy because when it's inadequate, it's associated with several pregnancy disorders that occur in the third trimester, including preeclampsia, which is a hypertensive disorder of pregnancy, and intrauterine growth restriction, which is when babies are born small. So to just give you an idea about the cellular processes that are going on during this remodeling process. So what we have here, the blue cells are trophoblasts, and these migrate down the arteries and actually mimic all the adhesion molecular repertoire of the endothelial cells. And this allows them to interdigitate into the endothelial cell layer around the edge of the artery. And once they're there, they actually induce endothelial cell apoptosis and smooth muscle cell apoptosis in combination with other cells in the uterus. And this remodels these arteries from these nicely structured, tightly coiled things you can see over here to completely remodel the vessel architecture here, including laying down fibrinoid deposits. So this is a really essential process for pregnancy. And most of this is occurring in the first 10 to 12 weeks of pregnancy. Okay, so where does shear stress come into all this? Well, we know that throughout the rest of the body, shear stress plays important roles in regulating endothelial cell homeostasis, vascular tone, and arterial remodeling. And the reason I was concerned about shear stress, not concerned, but interested in shear stress in the first trimester, is that these trophoblast plugs here actually block the arteries and prevent blood flow to the placental surface for about the first 10 to 12 weeks of pregnancy, at which point they dislodge and the blood can flow through to the placental surface. So that's a completely good and normal thing to happen in pregnancy. And actually the placenta happily exists without this blood flow during this time, and it's thought to be quite important. But what no one's really looked at is the fact that these plugs generate an increased resistive index and a turbulent flow in these arteries until 10 to 12 weeks of gestation. And we don't really understand the hemodynamic consequences on that process on spiral artery remodeling. So I wanted to look at two aspects of this. How does this low shear stress environment affect the migration of trophoblasts down the spiral arteries? And how does this low shear stress affect trophoblast-induced endothelial cell apoptosis once they have made it down the arteries? So we had a beautiful BioFlux in London that's set up at our lab here, and we had it sitting next to one of our time-lapse microscopes. So most of the assays I'll talk about today were all done in conjunction with time-lapse microscopy. And what I want to do in this talk is kind of give you a balance of the various different methods that I was able to use the BioFlux for to look at this problem, and also tie this into a story of what's happening in terms of uterine vasculature remodeling in the first trimester of pregnancy. So I'm going to talk you through the various co-culture models we used. The migration assays were developed looking for adhesion molecular expression in these cells following shear stress with immunohistochemistry on the plate, chemotaxis assays, and finally apoptosis assays. Okay, so I'll start out by talking you through how I seeded the co-cultures on the BioFlux. This is a pretty straightforward kind of thing, I guess, but when I've been talking to Mike, the slides are different to yesterday. Okay, I'll just talk. So when I've been talking to other BioFlux customers, this is one of the main things we end up talking about, is how do you get your cells seated beautifully on the plate so that you can do good experiments. So what I would do, I worked with endothelial cells, HUVEC cells, which I was able to isolate from umbilical cords a couple of times a week, which is one of the advantages of working in an obstetrics department, I guess. And I would seed these cells in at about 10 million cells per mil. So really, really high concentrations. And I would do this first thing in the morning usually. So put these cells in at really high concentrations, give them a chance to attach to the plate and spread out into what should be a beautiful confluent monolayer by lunchtime. So about a 4-hour incubation period for that. And then I would come in and seed on top of that trophoblast cells. So for our migration work, we mostly use a trophoblast cell line we had derived in the lab called SGH-PL4. And I would seed these at 7 million cells per mil, so just a 10-second pulse at 3 dyne per centimetre to get them into the channel. I would then allow these 3 hours to sort of attach down and spread out on top of the endothelial cells, and then run these on a time-lapse microscope for as long as the experiment required. I would seed these cells, sorry about this. So I would seed these cells on endothelial cells, and I would label them with different fluorescent markers in order to be able to differentiate the cells from each other. So here I've labeled HUVECs with mitotracker green, which we found to be best for their viability through the culture, and trophoblasts with cell tracker orange. Okay, so now I've got a little video which I'll just talk about while it uploads to you. So this demonstrates essentially just how these cells look on time-lapse, how they interact in culture. I'll point out a couple of cases where apoptosis is occurring, and just kind of give you a visual representation of what's happening. Okay, I think most of you have got it now. So you can see the cells move about in culture with each other. You can see instances of apoptosis, such as occurring where you get that bright membrane blebbing. And so this is actually run under 0.5 dyne per centimetre of shear stress. So not a massive amount of shear stress is flowing through there, which is why you see various instances of apoptosis. Okay, so I want to start by talking about our migration work. So the kind of line in our field has always been that trophoblasts migrate down the spiral arteries retrograde to flow. So they move against the blood flow, migrate down into the uterus. There's not a large amount of actual direct evidence for this. And so also when we think about it in terms of trophoblast plugs being in there and the shear stress is quite low in the first trimester, I wanted to sort of look at this process and maybe provide some direct evidence for it, or look at how it might be occurring and how shear stress may affect this process. So in order to look at migration, we actually worked together with Dr. Andreas Hopp and Dr. Daryl Greenhill at Kingston University of London, and we essentially developed a novel algorithm for automatically tracking the cells. So this allowed us to measure a large number of cells and a large number of sequences. I think we looked at 132 sequences and over 2,500 cells were tracked. So these guys are really, really technical at what they do. So I'm going to try and basically break down about 6 months of their life on a computer trying to build this algorithm into something that's basic and easy to understand. So I would give them time-lapse sequences with these dual-labeled cells, so green endothelial cells and red trophoblast cells. They would take each image in the time-lapse sequence, which are captured about every 15 minutes, split it into the red channel of trophoblasts. We're mostly interested in the end in trophoblast migration. They would apply a Gaussian filter to smooth this a bit, and then they would actually measure the migration of the cell, not just by tracking the general cell movement, but by applying vectors to every region in the cell that was showing up on fluorescence. This allows you to track the overall movement of the whole cell. If you've ever watched a cell move, it doesn't just move as one uniform blob, it sort of stretches out, it changes shape, it extends processes. This allowed us to track all of that movement within the cell. We wanted to look at cell migration in terms of how it was moving relative to the media flow in the BioFlux channel. So we assigned a compass point, compass directions to the cell movement from 0 round to 360. So 0 is moving in the direction of flow, and so 180 is directly retrograde to flow. And we kind of split up our analysis into, if you think of the compass in two semicircles, into those moving with flow, so 0 degrees and 90 other side of that, and those moving retrograde to flow, so 180 degrees and 90 other side of that. And each cell actually produced a distribution of values in terms of its directional migration, because all the vectors in the cell will sum together to produce sort of a peak. So it's not just moving in exactly one direction, it's moving over a range of directions, which are usually within a peak curve. Okay, so what we found from this. Along the bottom here, you can see the direction in degrees, and again, this is split up into the two regions I was talking about. Those retrograde to flow, and the two side panels are with flow. And have directional bias down the y-axis here. This is just an arbitrary marker that we've used. So here, for example, the 0.5 tiny blue line across here is essentially on baseline. So it's not showing any bias towards a higher directional bias or a lower directional bias. It essentially has no overall directional bias, either with or retrograde to flow. But you'll see as we move down an increase in shear stress, so 0.5, 2, 4, and 6 tiny, you start to get less directional bias in the region retrograde to flow and more directional bias in the region with flow. So this shows that rather than migrating retrograde to flow, as we previously believed, as shear stress is increased, trophoblasts begin to migrate with the direction of flow. And so we analyzed this statistically by averaging all the points in the retrograde and with area, and saw really quite significant differences between cells migrating with flow and cells migrating retrograde to flow. So many more cells going in the direction of flow. Now we ran these cultures both with trophoblasts cultured on top of UVEC and also with trophoblasts just cultured on the BioFlux channel, which we coated with fibronectin in each case. And there were really no major differences in terms of what happened with our cells when trophoblasts were cultured alone or when trophoblasts were in co-culture with endothelial cells. So you can see again the same significant difference at both 4 and 6 dyne and no significant differences at 0.5 or 2 dyne per centimetre. So this indicates perhaps that the ability of trophoblasts to migrate with flow at the slightly higher shear stresses isn't a result of cell interactions between the endothelial cell and the trophoblast, but it's something that trophoblasts will just do by themselves. So because we looked at vectors in each of the cells, this gave us information about not only direction but also speed. So we also looked at the speed data over the shear stresses between co-cultures and trophoblast-only cultures. There were no differences at all in the co-cultures from 0.5 to 6 dyne per centimetre. In the trophoblast cultures, there was one significant difference between 2 and 6 dyne per centimetre. However, that was the only difference we saw. There was no difference between 6 and 0.5, for example. So we're not sure entirely how much biological significance this finding actually has. I think overall, I consider this that shear stress doesn't really affect the speed of trophoblast migration. And I kind of liked this result because it indicates to me that because we've seen movement in the direction of flow, because there's no change in speed with that, it's not just a tumbleweed effect, if you will. So the cells aren't just being pushed along by the flow, and that's why we're seeing them move in that direction, because you would expect a corresponding increase in speed if that were going to be the case. Okay, so looking at immunohistochemistry using the BioFlux, we wanted to see if adhesion molecules played a role in the differences we see between 0.5 and 2 dyne where there's no migration and 4 and 6 dyne cultures where there was migration in the direction of flow. So we compared just a 2 dyne and 6 dyne culture, ran these cultures on the BioFlux, and then at the end of the shear stress run, fixed them in 4% paraformaldehyde, and then stored them in the fridge until we wanted to stain them. No, it's not going to give me the next section. Okay, so I'll just talk you through the method that we use. It's not going to show up. So I would fix them from the inlet well, but then when I went to stain, I would actually start working them from the outlet well, just, I think, for its proximity to the actual channel to make sure reagents get to the channel as quickly as possible. So I would work with about 150 microliters of volume in the channel, and whenever I added a reagent to the channel, I would pulse it through at 3 dyne per centimetre for 1 minute, and then I would trickle it through at 1 dyne per centimetre for a further hour or an hour and a half in the case of the primary antibodies. So we'd start by blocking the plate using 10% normal goat serum in that way, pulse that through at 3 dyne, and then run it through for an hour. Then just run some TBS through for 15 minutes at 2 dyne per centimetre, which is my standard sort of wash system. Then add 150 microliter of primary antibody in. Again, pulse for a minute at 3 dyne per centimetre, and then ran it through for an hour and a half, just at 1 dyne per centimetre. Wash with TBS, same thing, we'd use Alexaflor 488 secondary antibody. And then the final step we would do would be to add Hurst at a 1 in 250 dilution. And this was actually quite important in terms of ensuring that reagents could flow through the channel effectively. So because you're pushing your reagents through the BioFlux channel, and usually this works fine, every so often you get some small blockage and the flow is obstructed slightly. But by having the Hurst as the last step, if the Hurst has gone through and stained all your nuclei blue very nicely, you know there's been absolutely no issues with the flow through the channel at all, and all your reagents have got to the cells. So it's just a nice sort of control step as well as providing a useful contrast. Okay, so then we would examine these by confocal microscopy. So have the other half of the slide over. Okay, so these images just show you the kind of resolution that you can obtain. So trophoblasts here are again labeled red, so they're the sort of pinky red things, and the Hirsch counterstain is blue. We looked at 4 adhesion molecules, all known to be expressed by trophoblasts in the spiral arteries. And whilst this was nice for this presentation and gave us a lot of pretty pictures, when we went through and did blinded semi-quantitative analysis, we actually saw no differences in adhesion molecule expression at all between the different shear stress conditions, or in fact between trophoblasts cultured by themselves and trophoblasts cultured on HUVEC. So this is okay. Maybe this is in line with the fact that we saw no differences in directional migration between trophoblasts cultured alone and trophoblasts on HUVEC, so we didn't think that it was really going to be a result of cell-cell interactions in that. Also, maybe it's to do with the fact there was no difference in speed. So often we see adhesion molecule expression change when there's an increase in migration, and perhaps this ties more into the speed side of things, in which we saw no differences. Okay, so the next thing we wanted to look at was the fact that if we've shown that trophoblasts aren't migrating retrograde to flow, but are migrating with flow, how, in fact, are those trophoblasts then going down the arteries, and why are they going down the arteries? And so if it's not a result of them being stimulated to migrate retrograde to flow, maybe it's something that they're being attracted to down the arteries. So we used chemotaxis assays on the BioFlux to look at this. The 24-well BioFlux plate has 2 inlet channels that feed through to 1 outlet channel. So this allows you to put cytokine-containing media into one channel, control media into the second channel, and run these through at the same shear stress, so that this produces the same amount of force going down on both the inlet channels and results in parallel flow across the BioFlux channel. So you end up with half a channel with cytokine-containing media running across it, and half the channel with control media running across it. So we'd run these cultures at 2 dyne per centimetre for 12 hours. The thing we were looking at here wasn't actually the role of shear stress. There was just the mere movement of the trophoblasts between the shear stresses. And so capture time-lapse images every 15 minutes, and then just track the cell movement. So we would divide the channel in half. We would track all the trophoblasts on one side, which would be labeled fluorescently red, and track all the trophoblasts on the other side, and just record which half of the channel they ended up in. So then we could calculate the net migration into the cytokine-containing media just by taking the percentage of trophoblasts that migrated into the cytokine media and taking away the percentage of trophoblasts that migrated out of the cytokine-containing media. And so we looked at 3 different chemokines, which are known to be around the uterine environment, but are also known to be expressed by endothelial cells and show increased expression in low shear stress. And what we saw was that trophoblasts migrate towards all 3 of these chemokines, although this appeared to be quite concentration dependent. So in case of RANTS and MCP1, they only significantly migrated in 1 concentration used. But it was a start. And it kind of puts together the hypothesis that we started to work on, which is that, so we've shown if you've got shear stress and blood flow going up the artery, that would inhibit trophoblast migration down the artery. So the hypothesis we're working on is that perhaps trophoblast plugs in the first trimester prevent this inhibition, so they take it away. And this allows the trophoblast to migrate down the artery in response to other signals, such as chemokines such as these. And that's in fact how trophoblasts invade down the arteries in the first trimester of pregnancy. So these trophoblast plugs could be important in allowing this migration to happen. Okay, so the next section of work I looked at was what happens once the trophoblasts have migrated down the arteries. How does shear stress affect the apoptosis that's occurring? So we know that low shear stress increases endothelial cell apoptosis. So this is through downregulation of nitrogen oxide and the role of fast-fast ligand interactions. So we wanted to look at how this might play a role in trophoblast-induced endothelial cell apoptosis. So we ran our apoptosis assays on the BioFlux using time-lapse microscopy again. I've got here that we ran them at 0.5 and 3 dyne per centimetre. We actually ran them at 5 and 7 as well, to sort of take the shear stress up and see what happened as it increased. So again, we ran these over about 14 hours, capturing time-lapse images every 15 minutes. Now, the thing with apoptosis is that we wanted to really be able to run these experiments as long as we physically could to be able to get a really good idea of apoptosis occurring over time in culture. So the mechanism we actually developed on the BioFlux to do this was to take the plate off the BioFlux after 14 hours, replace fresh media in the inlet wells, take the media out of the outlet wells, and put it back on the microscope in the same position, probably adjust the focus slightly, and then run it again for a further 14 hours, and then track these two sequences together. So we just have an hour break in the middle. And that seemed to work pretty well most of the time. So we track apoptosis by looking for this characteristic morphology. So we just pick cells at the beginning, say 20 cells in a sequence, and visually follow them throughout the entire experiment and record when they die. So you can see this death by apoptosis here going phase bright in this membrane blibbing, and then finally complete cell death over here. I think time-lapse tracking of apoptosis is really a much better way than looking at endpoint apoptosis, say by tunnel at the end of the experiment, particularly in shear stress experiments where if your cell here has died, in the next frame, so these are captured 15 minutes apart, in the next frame that cell wouldn't be in the culture at all because the media flow through the culture would have completely taken it away, so that cell isn't going to show up in your endpoint analysis. Also, tracking cells in this way by time-lapse allows you to generate a kinetics curve of the death. So this allows you to analyze the area under the curve and look at what's actually happening in the culture. So this can still show quite a lot of differences even if all the cells in your culture end up dying by the end of the experiment. Okay, so what we found in these cultures was a couple of things. So here we have your lower shear stress cultures that were able to run for about 30 hours, and the two higher shear stress cultures that ran for about 13 hours. And I've just checked the percentage of apoptosis up the y-axis. And so you can see that, so I've looked at HUVEC apoptosis here, so we're looking at how trophoblasts kill HUVECs. And I've actually mapped a couple of different HUVEC populations. So firstly, we ran channels just with HUVECs by themselves, which are the blue lines which are kind of hiding under here. And then we had our co-culture channels. And I also looked at two different types of HUVECs in the co-culture channels. So I looked at HUVECs that were directly adjacent to a trophoblast. So in this case, they've actually used jar choriocarcinomas for our trophoblast cell line. So HUVECs directly adjacent to trophoblast and HUVECs more than 2 cells away from a trophoblast. So we did this in order to try and determine whether the apoptotic interactions were a result of direct cell-cell interactions or of things being released into the media, perhaps. And what we saw was that trophoblasts induced endothelial cell apoptosis in cells directly adjacent to them. So this green line here represents cells directly adjacent to trophoblast, whereas cells, HUVECs more than 2 cells away from a trophoblast, have the same level of death as control cells. And this is repeated again in a 3 dyne per centimetre culture. The second key thing we saw was that the ability of trophoblasts to induce endothelial cell apoptosis declines with increasing shear stress. So it's quite high, almost near the 40% mark in the 0.5 dyne per centimetre cultures, but this drops down as you increase the shear stress, so that by 5 dyne per centimetre you're really only getting about 5% cell death, and there's no significant difference between any of the types of HUVECs that we looked at. So this indicates that low shear stress promotes trophoblast-induced endothelial cell apoptosis. So we actually repeated this with primary isolated extravillous trophoblasts. So those trophoblasts that grow out of the placenta were able to digest early trimester placentas and use these cells in the BioFlux culture as well. And this showed the same effect of trophoblasts adjacent to huvics adjacent to trophoblasts, inducing more shear stress and reducing this as the shear stress increased. In fact, this effect was even more prominent when we used primary cells, which was very nice. Okay, so finally we wanted to look at why shear stress would affect apoptosis. We know from previous work that fast-fast ligand interactions are both involved in trophoblast-induced endothelial cell apoptosis in static cultures, but are also involved in the protective effect of shear stress against apoptosis in endothelial cells in the systemic circulation. So this is really always going to be our key target molecule of interest in this case. So we looked at this by adding fast ligand blocking antibodies into the media flowing through the BioFlux channel in our co-cultures. And so we looked at exactly the same two populations of trophoblasts, those adjacent, two populations of huvics, those adjacent to trophoblast and those more than 2 cells away from a trophoblast. And what we saw here, so this is just the green line you were looking at before, the huvics adjacent to trophoblast under control conditions or undergoing cell death. And when the fast ligand blocking antibodies were added, the amount of huvic death was taken right down to control levels. So fast ligand blocking antibodies did significantly inhibit trophoblast-induced endothelial cell apoptosis. Okay, so to try and tie all these ideas, I guess, into an overall model of what we're looking at. So we have these trophoblast plugs in the first trimester, and a lot of work has gone into looking at the fact that this prevents blood flow to the placenta, so what's happening to the placenta, it's in low oxygen conditions, and we know this affects various differentiation processes in the placenta. However, this is the first time that we've really looked at the other sort of branch, the other effect of these trophoblast plugs on the hemodynamic consequences. So we know that it increases spiralatory resistance. This is likely to lead to low shear stress. And what I've shown in this work is that this low shear stress has 2 ways to lead to successful spiralatory remodeling. So it provides permissive conditions for the trophoblasts to migrate towards the uterine chemotactic factors. And it also provides trophoblasts with a survival advantage over endothelial cells, which is data I didn't get a chance to show you today. And trophoblasts have an increased ability to induce endothelial cell apoptosis. So all of these things combined lead to successful spiralatory remodeling. So finally, I'd just like to thank everyone that's contributed to this work and contributed to the funding of this work. In particular, Judith Cartwright and Guy Whitley at St George's, who are my main mentors there. Thank you.